Perfect.
Okay. All right. We can get started. All right. Welcome, guys. Thank you for coming in and joining us today. Today we have a very exciting presentation from Dr. Knock. Many of you guys have probably seen Dr. Knock on TikTok. If not, be sure to follow him on TikTok or Instagram. It's at dr.knock. That's right on the screen. A little bit about Dr. Knock, and then I'll let Dr. Knock introduce himself. Currently, excuse me. Currently, Dr. Knock is a PhD scientist. He's working in drug development and pharmaceutical sciences. He's also working in developing therapeutics for COVID-19. So very exciting stuff. So I'll let Dr. Knock introduce himself.
Okay, great. Yep, thanks for having me. You covered it well. PhD scientist working on developing therapeutics for COVID-19. And so we're going to talk about three main things today. First of all, what it's like to get a PhD or to have a PhD, the types of jobs you can do with it. It's the similar type of fundamental training in undergrad to being pre-med. So this is one additional example of things you can do otherwise with it. And then we'll talk a bit about COVID, particularly focused around the theme of the mistake of thinking in binary for COVID-19, which is how a lot of people get an overly simplified view of COVID, which ends up being totally wrong, which can be either dangerous or can make it seem like scientists don't know what they're doing. We'll talk through a couple examples of that. And then third, we'll talk about just some best practices for doing health and science outreach on social media. Basically just some tips I've learned pretty much over the course of the pandemic, which is when I started my account. That you can implement even just on your private accounts. If you're making videos for your friends and your family, even if you never share them publicly, some things that you could start to do. Okay, so first though, starting with the PhD, or rather about me a bit more. So in undergrad, I studied biomedical sciences. This was a combination of molecular biology and microbiology. It was basically the major that everyone did if you were pre-health in some form, or if you were pre-graduate school in some form. And so I took a lot of courses in like micro and immunology and cell and molecular biology, which is just a good foundation in, small scale science. I minored in cognitive psychology, which was very interesting, which I haven't used very much. Maybe it's influenced how I think about stuff, but it was fun. Graduate school, the division I was in was called the Division of Pharmacoengineering and Molecular Pharmaceutics, which is a very complex name. I'm personally pretty convinced that we ended up changing it to that just to make it sound impressive on business cards. But what the division does or did was drug delivery. And so that's where someone has made a really great drug for cancer or something. And it works really well at killing tumors, but it's also very toxic. And so sometimes your goal is to put it into a nanoparticle or to conjugate it to some type of a protein that helps you deliver that drug where you want it to go in the body. Or maybe you're like designing capsules for oral drugs or something. That's sort of the field of molecular pharmaceutics and drug delivery. Specifically, what I was studying was how to prevent unwanted immune responses against drugs, which can happen. And it's not good when those happen because it's basically like having an allergy against the medicine that you're taking. When that happens, you both clear it rapidly from your system, which means the drug can't do whatever it was supposed to do. It also puts you at increased risk of like going into anaphylaxis when you take that medication, both of which are not good. So I was studying ways to prevent that from happening with one class of therapeutics. Current role, Assistant Director of Development. So I'm working on treatments for respiratory infectious disease. This was true even prior to the pandemic. We were working on things like influenza, RSV, MPV, a couple other viruses. And so it was pretty easy to just switch over from those that we were working on in the same antibody-based platform and swap in COVID-19. And so now we are additionally making antibodies for COVID. Specifically, my role is in product development from preclinical to clinical. We'll talk about that a bit on the next slide. I also do some scientific and management consulting with companies who are looking for help with, things like we have a device that we may be interested in turning into a medical device. What are the steps we have to go through to meet FDA regulations to get that device approved, as well as more broadly management consulting, where, even companies who have no real scientific goals, just looking to have a question answered with the scientific method. And so I can help out with that as well. Then, of course, I do outreach at Dr. Knock on TikTok and Instagram, and now on YouTube. I do have a YouTube account. I don't use it too much. Okay, but here, what is development in biotech? This is my role. And so what's shown here on the right is unrelated pretty art. What's shown on the left is the various phases of clinical and preclinical development. So stage one here in the purple, I don't know if you can see my mouse. Basic research is like you discover a cool molecule. Early discovery is you find that molecule binds well to some target in the body that is a common target for diseases or something. And so when you have those hits, you can test them then in preclinical studies, which is usually the phases of animal testing, both for efficacy and for safety. And then you have to produce basically a big, extremely long packet that you have to give to the FDA. And then you say, we think we're ready to go into humans. That application is called the IND. It's the Investigational New Drug application. And then you're in clinical development. This process takes a very long time. But my job is mostly focused around these two, the early discovery, the preclinical, and now we're just starting to get into clinical development with some of our COVID therapeutics. So I take a look at what the FDA requirements are. They publish these very long lists of the things that you need to do with your drug development program before you can even consider going into humans. And so then I help manage the experiments, to plan the experiments, to make sure we'll get the data we need to submit it to the FDA eventually. And then, that can range. We'll go through an example of like a typical day in my schedule, using today as an example, actually. But it can range from planning actual experimental steps to analyzing data to putting together proposals for companies that we may work with. So here's today. 6:30, got up, read a paper, like an academic journal article. Pretty important to keep reading those. Otherwise, you very quickly fall behind in the world of research. Get ready, take a shower, have breakfast, plan a TikTok. Then from 8 to 9 is usually about the range where I'll record a TikTok and then start to answer emails, both for social media stuff as well as for work stuff. Then had a meeting today from 9 to 10:30. This was an internal meeting within our company to discuss some data that we recently got, where some experiments did not work out as well as we had hoped they did. So like things that we're going to do next time to try to make it work better. 10:30 to about 11:30, I was writing a protocol we're actually testing a new drug, not for COVID, for a different disease in an upcoming study. And so when we do experiments, we pre-write out every step of the protocol ahead of time so that there's no question of if it doesn't work, it's because the actual drug wasn't working. It's not because we messed up the protocol because we didn't have it written down very well or something like that. Today was fun. 11:30 to 12, I was actually on the radio with a channel in Ontario, 900 ZHML, talking about COVID-19 vaccines, which we can talk about a bit more later. Then I had lunch. Then I analyzed some data from ELISAs, which is analyzing antibodies, in this case against COVID, after we had nebulized them into a form of drug delivery. Then from 1:30 to 3, I was working on editing a draft of a manuscript that we're going to be submitting about COVID antibodies to a journal. Okay, then 3 to 4, had another meeting. This one was not within our company. This was within a third-party company who's helping us execute some of our studies. We're testing an anti-influenza drug, and we did a run of that experiment, and none of the animals actually got influenza at all, including the ones that were supposed to, like the control animals. So we have to redo it. And so we're talking about how to make that work better next time, and then analyze some more data, then went on a jog, took a shower, made dinner, browsed TikTok, did some consulting. 8 p.m., now we're here. And then after this, I'm going to hang out with my wife. So that's pretty typical. It's a good split between analyzing data, planning experiments, working with third-party companies to execute studies or plan studies. And that is sort of my role in development. Here, I will say, if you guys have any questions, just make sure you post them in the chat, and we can either go through them as they pop up, or we can talk through more questions at the end.
All right, just quickly, I see a lot of questions in the chat about summary, which is a way for us to certify your attendance. I can, since there's a lot of question about it, I can go at the end of the call. I can address that real quick and regarding what goes in a summary and all the instructions for it. Okay. So far, no questions. We're all good. You can continue.
Okay. Okay, so these are some of the common questions that people don't really know often. I didn't even learn these in undergrad until I was in basically my second or third year. It's often you'll have a very good idea of what the pre-med path looks like, what it takes to get into med school, what you need in your application. It's less clear. There's fewer resources on what it takes to do a PhD or what it's like to do a PhD. And so that's part of the point today, is to teach you about that. I was pre-med myself, actually, for the first, I guess, year and a half of undergrad, until I tried out doing research in an actual research lab, which is totally different from, in a class lab, like where you go to organic chemistry lab or something and do the preset experiments like a real research lab. So who knows, you may end up changing. But some common questions here. How much does it cost? Usually you do have to pay for the application fees for PhD programs, which they can be waived. There's hardship waivers at pretty much every school. But then they pay for your flight and your hotel and your food and everything while you're at the PhD interviews. The actual PhD itself also does not cost anything. They actually pay you. So they pay your tuition. They often pay like your health insurance. You'll additionally get a stipend in the range of $20,000 to $30,000, kind of depending on where you live or what you're studying. And so the reason for that is because you're basically working full time when you're getting your PhD. What makes you a competitive applicant for grad school? The big one is research experience. And the reason for that is you have to demonstrate to applications committees that you know what it's like to do research, which is not what it's like to do a lab for a class. It's the same thing with med school applications. They're making an investment when they choose you to come to the program. And they want to know that you know that it's going to be really hard, that what the career is like when you actually get into it. And so for the PhD perspective, research, you may do experiments for 2 months in a row and they just repeatedly fail every week. And so admissions committees need to know that you know that that happens and you're okay with it and you're resilient and you're inquisitive, you're enthusiastic about whatever area of science you're interested in. How long does a PhD take? It's a pretty wide range. There's no set number of years. It's typically in the range of 4 to 5 or 6 or 7 years or so. I think the average is in the range of 5 years. Do you have to get a master's first? You do not. You can go directly from bachelor's right into PhD program. In your first couple years of your PhD, you take courses for maybe 10 hours a week, and then you're in the lab doing research the rest of the time. And so that's sort of like a built-in master's as part of the program. We'll skip over how do you choose a research lab. That's more relevant like in your first year of grad school. What do you actually do during a PhD is a common one. And so basically that first year, you're only doing 10 hours of classes per week. The other 30 or 40 hours, or potentially more, you're spending in the lab. And so you have your own projects, which you're shaping into your thesis. Like mine was about those immune responses against drugs. And you're just doing experiments pretty much full time the rest of the time when you're not in courses. You're generating data, which goes on to get grants, which brings in a lot of money for the university, which is why they pay you to get a PhD. You're basically working to provide these things that are bringing in money as well. Who should consider a combined MD PhD? That we can talk about at the end if anyone has questions. Very few people is my answer. And no one should consider it simply for the purpose of when you do an MD PhD, they'll often pay your medical school tuition as well, in addition to the PhD tuition. That's an extremely bad reason to do an MD PhD, which we can talk about if anyone's interested. And then once you have your PhD, what types of jobs can you get is an important question, because you probably have, like you may have had professors or teachers who have PhDs. That's not the only thing you can do. That's the image you may have in your mind, but there's really quite a lot more, right? Ranging from regulatory affairs, which is, making sure that a company is in line with all of the regulations that they're supposed to be, so they don't get shut down by the government. That's typically a PhD level role, particularly in science-based companies. Medical writing and technical writing is a huge field. And so that can extend to scientific writing with a PhD. And so there's a constant need for people to take complex scientific and medical information and distill it down into a form for press releases for the general public or for investors, or to take foreign authors, maybe they do research in Italy and they're experts in the field, but their English is not very good. And so they may write a paper and then they need to pay someone to take it from decent English into like professional American scientific English. Huge market for that. Business development is sort of applying, some of the scientific skills in project management. When you have 5 experiments going on at once, you develop a lot of skills that can be very marketable in the business world, even if you're not working in science. Technical marketing, medical science liaison is like marketing drugs. Basically, it's taking pharmaceutical company results and showing them to the clinical community. Basically to educate healthcare providers, like if you got really cool data from a phase 3 clinical trial, but no one knows about it, no one can prescribe your drug to help people or to sell more of your drug from the perspective of the company. And so medical science liaisons help teach people about new drug trial results. There's policy positions in the government. There's consulting outside of science, which I've done some of. You can obviously work as a scientist. You can work as a professor. You can work as a professor in a lab at a university, like a research lab. You can work for contract research organizations or manufacturing organizations, which is, you may think of like pharmaceutical companies as it's this big unit that conducts research. And if the research works, they sell their drug, or they may be a makeup company and they're looking for the exact right formulation of cream to put on your face. That is a scientist, that's a division of scientists who are doing those types of jobs. But there's also third-party companies who you can pay, who specialize in like making pharmaceutical grade creams for your face. And so that may be a contract research organization where if you're a wannabe makeup company, you can pay them, they can help you develop your cream, and then they bring it back in-house, strap your label on it, and you can sell that. So all of these things require scientists, and most of them are not at a university setting. I think a lot of people don't realize that.
So pretty broad in terms of how you can go in if you want to get a PhD. There's a lot of fields you can go into if that's something that you're interested in. There's a question I think that ties in well. It's from Javier and Michelle. I can merge them together. Like, how much research experience should you have prior to going into grad school? And like Javier asked, like, what are ways that you networked with professors or doctors to get those research opportunities in college? Like, how should you go about getting research?
That's an extremely good question, which I was not already planning to cover. So this is good. The amount of experience that I had was not a lot of experience. I started after my second year, basically at the beginning of my third year. And so by the time I had applied, I had maybe just over a year of research experience working in a lab. You certainly do not need 3.5 years of research experience to be competitive for graduate school. What you really need is to show that you have, that when you did your research, that you were playing an active role in it. And so how you describe your role in a research lab is almost as important as what you did while you were in there. And so, talking about not just, I was helping out with them making bacterial plates or something. You want to say what the objective was of your research, what the specific role you played was, what you learned from it, and how you think that makes you competitive for graduate school or something. And if you have all of those elements in every type of research experience you have, that helps convince admissions committees that you have an idea of what it's like to work in research. You've worked with a graduate student or with a professor in much the same capacity as like an early graduate student would. Now, how to get a research position in a lab is extremely difficult right now, obviously, because a lot of labs are operating at very reduced hours. Some general principles apply. There's a very incorrect way to approach a professor for a research position, and there's a very correct way to do it. The very incorrect way is to send a copy-pasted email to 20 different professors saying, I'm a pre-med student, I'm interested in research, do you have a spot in your lab or something? No one is going to basically read that email. If you want to have even a shot at getting your foot in the door, you have to demonstrate, like from the very first contact with that email, that you know what they're studying, that you've made an effort to read at least one of their recent papers. It'll take you some time. It'll probably take you an hour to do, but you should be able to come up with a question about their research, like, this is really interesting. I found this to be particularly fascinating. Where are you going from here with this research? Or what's the current status of this project? If you ask questions like that, your chances of getting a response back from a professor, they're probably like 5-fold higher or 6-fold, who knows?
It shows that you're interested in what they're doing, not just getting into any research lab. It shows that you care about what the research going on, you understand before you come in.
Yeah. Exactly. Because if they think you've spammed an email to the entire department, they get dozens and dozens of undergraduate students who want to do research. And so it's only the ones that very obviously want to participate a lot in the research lab who are going to get it. Because it's a big investment to bring a new student into the lab. It probably takes at least 3 or 4 months of dedicated training before like you're independently productive, I would say. So you have to make it very clear that you're passionate, you're going to stick around for at least a year. Don't say you'll leave after a year, but make it clear like you want to do this. That'll help a lot.
Good. All right. Thank you for answering. You can continue.
Yeah, good questions. So this is sort of the point that you were making in the breadth of what you can do with science. Science is really, it's a skill set, right? There is some knowledge, like I have a lot of background knowledge in several key areas. But that's not really what's marketable about being a scientist. What's marketable is the skills that you've gained over the course of your training and your research, right? Summarizing complex information in a way that's understandable by whatever audience you're presenting it to. Being able to independently get up to speed in new projects. being able to analytically design experiments in a research setting, that's very applicable to a lot of things in the business world, right? Where you're testing this marketing approach versus that marketing approach. How do you design the experiment to see which one is actually better than the other? Persuasive writing for publishing manuscripts and stuff. Working well as part of a team. All these things are crucial to being effective in the science world. They're also crucial to being very effective in the business world. And so once you have that skill set, and interpersonal communications should be on here as well. Once you have that skill set, you certainly can go work as a scientist in developing facial creams or toothpaste or drugs or whatever it is. But there's a lot else you can do. And so in some sense, you're only limited by your creativity when you're thinking outside of the box there, because most, maybe all industries could benefit from PhD level, like analytical analysis of whatever business process they're doing. But it is hard. It's hard to sort of come up with those niches. One thing at the bottom there that I highlighted, within science and academia, scientific training is the norm. Having a bachelor's, master's, PhD, like even having a PhD is not that special if you're working in a drug company where your entire division has PhDs basically for the most part. Once you step outside of that environment though, all of a sudden your PhD is not everyone has a PhD in the vast majority of the business world. And so there's some inherent value there, which can be marketable if you're clever about doing so. Okay, so now we're going to swap. We'll talk briefly about some topics in COVID, just because I figured people would be interested in some general COVID stuff as well. This is how this sort of broad principle of thinking in binary is how people get a lot of stuff wrong about COVID-19. And we'll go through some examples of this. Basically, this can lead to, and is the direct source for a lot of occasions we've seen over the past few months where people think, scientists are flip-flopping on an issue, or they have no idea what they're doing or something. In reality, that's often because people are thinking that scientists are going from like 0 to 1 in these binary decisions, but almost everything is a gradient when it comes to COVID-19. This is a good example. This is what made me think of doing this segment. I was doing an interview with Brute a couple of weeks ago, and the interviewer asked me, which is more effective? Is it masks or is it social distancing? And so when I first started to answer, I was trying to actually think of what the answer to that question is, because there is an answer to that question. If you're going to do one or the other, which one is more effective? But I shortly realized that's not how you should answer the question. The real answer is that both of them are far more effective than either one alone. And so this is part of what's called the Swiss cheese model of risk reduction. Whoa, did not mean to scroll. Okay, Swiss cheese model of risk reduction here. If you look at each slice of Swiss cheese, you may have seen this analogy before. Each slice of Swiss cheese has holes in it, right? You can put a respiratory droplet right through any given slice of Swiss cheese. But when you stack a bunch of slices of Swiss cheese together, you can't get through the whole block. And so that is how you should be thinking about, it's not masks or social distancing. It's if we combine masks, social distancing, hand washing, meeting outdoors, or having good ventilation, right? The combination of those, even if they're each only 60% effective at reducing your dose of virus that you emit or receive, when you multiply 0.6 times 0.6 times 0.6 times 0.6, it very quickly brings risk down to an almost inappreciably small number. Here's another one, infection events. Not a binary thing. It's not that you get infected or don't get infected. In most animal models of respiratory disease that we use if we're testing new drugs for respiratory disease, for example, COVID here, higher doses of virus leads to higher risk of more severe disease. Maybe that's not surprising, but it's underappreciated, I think, in that when you're thinking about what's shown on the right here is some data with hamsters where if you expose hamsters, a typical model of how sick they're getting is how much they're changing in weight. You expect them to grow over time. If they're infected and they're having severe disease, they can lose weight. And so MOC means these were not infected. The black line, that's like a healthy amount of weight gain. What you see though is that the group that received a high dose of COVID-19 versus a low dose of COVID-19, the high dose group lost more weight. And so that probably doesn't seem very surprising, but this is not discussed really, in that all of these measures that you're taking, each slice of Swiss cheese, your goal isn't necessarily to stop 100% of the viruses from being inhaled. Your goal is just to minimize the number as much as possible. Because even if you still get infected, and the minimum number to probably cause an infection, we're not really sure, it's probably in the range of 100 or a couple hundred virions. But there's a big difference between me inhaling right now 200 viruses or 200,000 viruses. And I just think that more people should think about it like that. Here's another one. COVID-19 outcomes, recoveries versus deaths. You'll see people quoting survival rates for COVID-19 as like, well, if the survival rate is 99.8% or whatever, I just made that number up. Why do we have to be concerned? And part of the answer is that it's not you survive, it's not you die or you're totally healthy, right? There's a large fraction of people who recover from COVID-19 who have potential long-term adverse consequences, right? We see pulmonary fibrosis, which is lung scarring, particularly in people who have had severe disease. We see that with COVID-19. We saw the same thing back with SARS. We're seeing cardiovascular complications, even in people who have relatively mild COVID-19. It is actually infecting the heart. There's evidence of heart damage. It's unclear how much that'll predispose to increased risk of cardiac disease moving forward, but it definitely seems like it could. Neurological disease, right? COVID it's partially a vascular disease, and your supply of blood to your brain is one of the best predictors of, for example, like your cardiovascular health in middle age is one of the best predictors of your risk of dementia in older age. A healthier blood supply to your brain reduces your risk of neurodegenerative disease. So in COVID-19, if you have vascular damage and you have potential infection in some neural components, like I was talking about smell in a recent TikTok, who knows what the very long-term complications of a COVID-19 infection will be? And so to look at the COVID-19 survival rates and say, well, only 0.2% of people die, that's not capturing the fact that some other percent, and we don't have a good idea of that percent, maybe it's 2%, 3%, 4% more people have these potentially long-term consequences.
So, right, like you said, even though the survival rate is very, very high, and I think many people take that for granted in a way, like, I'm young, I'm not gonna, even if I get it, I'll be fine, so why wear the mask, whatever. But like you said, we don't know a lot about the disease itself, and there are also long-term problems that we might not even know now, or the ones that you pointed out could go in the long run and affect you. So it's better to not have it to begin with.
Yeah, that's exactly it. It's the precautionary principle of, we don't really know what's going to happen long-term with COVID-19 infection. So I'm wearing a mask now for sure. And even if a mask doesn't totally protect me, if it's a cloth mask or a surgical mask, maybe it does reduce the virus I inhale by like 10% or something. I'll still get infected maybe, but a lower dose maybe reduces your risk of some of these other complications as well. Ooh, here's another interesting one. So a lot of the graphics that are done for COVID related stuff are extremely wrong in what they represent biologically. And so I took this from an article about aerosols, I think the one on the left, where it shows, 2 viruses or 1 virus per droplet of liquid. That is not at all close to the typical size range of droplets that we actually emit. What's much closer is the one on the right. And so there's a lot of misinformation about the fact that, well, the mask only filters out particles larger than 0.3 microns or something. And the virus itself is smaller than 0.3 microns. Therefore, there's no way that masks could filter out viruses, which may be true if viruses were moving through the air as naked, just the virus going, but that's not how they work. They're in these droplets of water, mucus, and protein, pretty much close to the size scale on the right here, where the virus itself may not get through or may go right through a mask, but this entire droplet is not going to go through the mask nearly as easily. And so part of the reason I think why this misunderstanding persists is that we see art like the one on the left here, which looks a lot better than the one on the right, which is totally wrong. And it makes it look like the droplets are only a little bit bigger than the viruses. And so maybe they can get through the masks or whatever. And I don't really have a suggestion for this other than you have scientists making the art at the same time. This is another example, though, where it's important for scientists. We talked about medical communication, medical writing. There's also medical illustrating and graphics, right? If you're looking to communicate science and health information, one very effective way to do it is make videos and make Instagram story posts for big accounts and stuff. And so posting one on the left is going to maybe harm public health perception. And so you need scientists involved who are intimately familiar with the size scales at play and like the real scientific mechanisms at play. Community immunity. We're going to skip past this slide. This is what we're going to cover though, because this is directly related to the vaccines and our chance of herd immunity. And this is sort of how some of the calculations work. This is fictitious, which I'll show you on the next slide, but it's a good model for understanding how to do these types of calculations. So community immunity, also known as herd immunity, but we prefer community immunity because it makes you sound not like an animal, even though it's a little bit harder to say. Basically, you prevent the spread of virus when each infected person spreads it from themselves to only one other person. Ideally, they spread it to slightly fewer than one other person, so it slowly goes down over time. But as long as it's just one person that you're giving it to, at least it's not growing in terms of this exponential spread of disease. So the threshold you need to hit in terms of community immunity to make that happen depends on the average number of infections that each person causes. So if each infected person typically infects 2 people, this is just an example. I'm not entirely sure what the number is for COVID. I think it's closer to 3, but just using 2 as an example. The number of people who have to be immune has to be, the formula is up here. It's 1 minus 1 over R naught. R naught in this case is 2. That's the average number of people each person infects. So 50% of the population would have to be immune, because now instead of, I don't know if you can see, I'm probably pretty small on the screen, but instead of 2 people being infected, it's only 1, so 50%. The same calculation if R naught was instead 3 people who typically get infected. In order for only one of those to be a productive infection, 2 of them have to be immune, which means you need a 67% immunity rate in the population. And so what we're dealing with COVID is probably closer to that second example. We'll probably need close to 70% or so immunity in order to basically stop transmission. So the way I've drawn it here is conceptually useful for understanding how vaccination works. It's not at all how spread actually happens. I'll show you how it actually happens on the next slide. There's a lot of heterogeneity. And so we talked about R naught, that's the average number of people who get infected. This other variable, it's a variable called K, is the measure of how heterogeneous it is. Is it everyone infects 3 people? Or do some people infect 1 person, some people infect 10 people? And it's the latter case for sure. This is much more close to what it actually looks like with COVID-19. You've got this one initial infected person over here. This person may infect 7 people, or however many that is. And then each of those 7 people, this person infects no one. This person infects no one. This person infects no one. This person infects 1 person who infects 2 people, one of which infects 5 people, right? Most of the trails of COVID-19 are probably going to end in a dead end or infecting only 1 other person. And so where you see a lot of the spread coming from are these relatively rare, but relatively super spreadery type of people. And so this is where you see, there's a group of 50 people who are inside a room with poor ventilation singing or something. And then one person infects 50 other people, probably through aerosol. That seems to be a relatively dominant mode of transmission. I think the estimate, it's close to 15 to 20% of people who are infected with COVID-19 are responsible for causing upwards of 80% of all the new infections, which is hard to deal with conceptually. What it does mean is you have to weigh the relative value of these 2 different people, or vaccinating these 2 different people shown here. There's an inherent difference here where vaccinating this person, it may protect them from being infected, but they weren't going to infect anyone else anyway. In contrast, this person over here, if you vaccinate them, you prevent these 5 infections and who knows how many infections downstream. Unfortunately, we can't really use this information because you can't selectively vaccinate. You don't know who's going to be spreading it to more people, because part of it is behavioral in going to parties and not wearing masks and not distancing and stuff. But part of it is biological. There's large differences across people just in the amount of virus that they produce while they're talking and while they're breathing. We don't really understand why there's such big differences in how much virus you're shedding, but there are, particularly for COVID-19. Okay, here's a common question. Does the vaccine mean the end of the pandemic? Unfortunately, the answer is no. It's going to be a gradient, right? It's not going to be situation A here. It's not going to be, we roll out the vaccine in January, February, pandemic is over, stop wearing masks, stop distancing. Mostly because in this case, there's going to be such a limited supply. And so what we're going to see is rolling out the doses of vaccine to the high risk people, which is not going to stop transmission in the community like we just talked about. You probably need 70% of the community to be immune before you start to slow down transmission. And so what we're going to see then is high risk people will be removed from the transmission pool. And so the mortality rate will start to come down within a couple weeks of, or probably within a month or month and a half of when vaccines start to be rolled out. And it's going to keep going down as more people get vaccinated. And eventually that will also coincide with decreased transmission as larger percentages of the population become immune. How was the vaccine developed so fast? We can talk about that if people have specific questions. That's a very common point of hesitance in terms of maybe being willing or unwilling to take the vaccine. I
I have a question, and there was a question about the chat too. So right now,
Okay.
we're in the first wave, and the cases that recently, for those who are following, I follow, it's north of 100,000 cases a day. So that still
Yes.
continues into the first wave. How do you think heading into the winter? Do you think we'll get like a second wave, or like with the vaccine coming in, will there be a second wave after?
It's a good question. Yeah, describing the waves, you could even potentially say that we're currently in our third wave of COVID-19. If you're counting the peaks and the troughs, it never came to baseline. It never went to zero. I'm not really sure it is going to go to zero. And so if your question is, we're spiking hugely now, that's going to continue for the next couple of months. Next couple of months are going to be rough. We don't have the vaccine yet. We have some better therapeutic options, which means, mortality is going to go down. But we're in for a difficult couple of months. However, once we get through the next couple of months, I do think it is going to get progressively better. We're going to peak, and then we're going to implement more restrictive measures like we have done the last two times. And then cases are going to start coming down again. And then we're going to start vaccinating people around that time. And then we're going to start releasing some measures. And then cases are going to start to come up again. But on that next one, the cases are definitely going to come up again after that. But the mortality rate is going to be slower to come up because at that time, the difference is we have been vaccinating those high risk people.
Got it.
And you said there was a question from the chat, or was that?
Yeah, that was a question from the chat and mine too, but
Okay.
there was another question. Will there be, we talked about this earlier, but the vaccine that's out from Pfizer and Moderna showing 95% success rate. There's a question, what is the likelihood that the first set of vaccines that will come out will have complications to the people that receive it?
Yeah, that's a great question. So what about the long-term effects of a vaccine? What are the potential complications?
Yeah.
This is, and I need to make like an actual video, like a TikTok and Instagram video about this, because this is probably the most common question people have right now. Like, the vaccine works, it is effective at preventing disease, but what is going to happen in terms of side effects? And there's a little bit of nuance to thinking about this. The comparison is not getting a vaccine versus not getting a vaccine. It's getting a vaccine versus potentially getting COVID-19. And so, we talked about earlier some of the long-term effects of COVID-19 can be debilitating. It can cause death in a small fraction of people. Who knows how bad it's going to be? Some people have long hauler COVID for 5 or 6 months, right? They see decreased exercise capacity ever since they've had it, basically. And so in comparison, when we look at the vaccine, we don't see any of this happening. We see people have, injection site soreness or swelling. They may have a headache. They may have fatigue or fever for a few days, but then it goes away after the first week. And so the two main adverse effects you're worried about with vaccines are, one is called antibody-dependent enhancement, which is where it can happen where having antibodies against a pathogen actually makes your infection worse the next time you get infected. And that it depends on the type of virus. It happens, for example, for dengue virus. And so who knows if that can happen with a new virus? It's something you have to check out. The time that you would see that happening, though, is the next time you get infected. And so that would be captured in the clinical trial. If antibody-dependent enhancement was happening, what you would see was that when you're comparing the vaccinated group against the placebo group, the vaccinated group actually has worse outcomes than the placebo group. And because the antibodies are helping facilitate entry into macrophages or something, that's very uncommon for vaccines, but it can happen. That's why we're running a clinical trial. So that's thing number one. Thing number two is Guillain-Barré. And so that is a separate type of inflammatory response that can happen to vaccination or to infection. And so it can happen, for example, to the flu vaccine. It can also happen when you're infected with influenza. And if you compare the rates of getting Guillain-Barré syndrome with actual influenza infection versus the rate with the vaccine with the vaccine is much lower than it is with an influenza infection. But that is a concern, because what if it is actually higher than the rate that's seen after infection? But the typical time of onset of Guillain-Barré it's usually not greater than, the scale of months, certainly captured within the scale of the clinical trial, I guess is what I'm trying to say. When you think longer term than that, people are like, well, what if the vaccines cause cancer or something? And there's no plausible biological mechanism by which they would, is the answer. We have a broad history of vaccine development experience and watching what happens when populations get vaccinated. what happens is rates of infection go down because the people are having immune responses against those diseases. We don't see any correlation with cancer rates or autoimmune disease or autism or any long-term chronic diseases broadly following vaccination. And so we have to make the benefit risk judgment call of, if you were to bet your money on which one has the higher the higher chance of severe long-term effects, is it a COVID infection or is it a COVID vaccine? Like, it's definitely the COVID infection presents much greater risks of these potential long-term effects. And so that's sort of where we're at.
Okay. Thank you for explaining through that. So taking the vaccine is a better shot than not taking, because there's no evidence of a vaccine causing an autoimmune disease or autism or cancer or any of that stuff. So
Right.
we're clearing that. There's a question from Carla. At what point I think you addressed that. At what point should we feel like the vaccine is safe? That would be more of a long-term thing as more and more comes out. But there was another question. Would you take the vaccine if it was approved by the FDA?
Oh yeah, I would get it. I would honestly probably get it like right now if it was available to me. It won't be available to me right now because I'm not in a high-risk group. But as soon as it is available to me, assuming that there's, this is the preliminary data report that gets released. Carla's right to ask. You should wait until the final FDA approval. Assuming that happens in the order of a couple of weeks, I am definitely on board for sure.
Yeah.
Because I'm scared of these things on the screen. I don't want pulmonary fibrosis. I don't want vascular damage. I don't want neurologic disease. I don't want cardiovascular damage from a COVID-19 infection. I'd rather take the vaccine and have fever for 2 days. Okay. So the last part here, and this is a relatively short part, is scientific communication. This is 2 quotes. I had made the slide actually prior to the most recent vaccine data, but it's still relevant. These are 2 quotes. One is, "We are not expecting to see widespread vaccination until the middle of next year." There's a quote from Margaret from the WHO, versus, "The vaccines are coming momentarily," from Donald Trump. And so my question generally is, if you're a member of the public, how do you interpret these apparently contrasting statements, right? And the answer is that you think that one of them has to be wrong, basically. One of them does not have to be wrong in this case. It depends on what fraction of the vaccine development cycle you're looking at. Like right now, you could say that we have a vaccine or we have 2 vaccines. But so in that case, Trump, is referring to that part of the timeline. But that's very different from me being able to go to the pharmacy and get a COVID-19 vaccine, right? That's probably the middle of next year. But the difficulty is that that's not always clear in these presentations, right? And so Trump comes out afterwards and says, like, "See, I was right, we've got a vaccine." But the other person wasn't wrong in this case. And some of that nuance just is not communicated. And so that ties into science medical communication. Presumably, most of the people watching this video are part of the scientific or medical community or would like to be at some point. And one thing that I strongly believe is it does not matter how much we know, for the most part, how much I know, how much you know, how much anyone watching knows about science and medicine, unless you're able to communicate it to other people effectively, right? You can be the best doctor in the world and know, all the best things to do in terms of lifestyle and drugs to treat whatever diseases. If you can't effectively communicate that to people, you're not going to be a very good doctor. And so it's important for everyone to begin to practice, and this is also true in science, right? If you make cool scientific findings, you have to be able to tell people about them for them to be able to make use of them and do other cool discoveries. So I think it's important for everyone, basically as part of your training, to get good at communicating scientific information. And the only way to do that is to practice. You're going to be awful at it at first. I'll skip over. No, I'll do this slide. So if you were to weigh the potential value to society, to people's health, due to the implementation of the science and medicine that we already do know right now, versus what could be gained by developing new knowledge or new medications or treatments for cancer or whatever. I didn't think about this until I went to a seminar and someone brought up this point that we have an extreme failure to apply a lot of things that we know would make people a lot healthier and a lot happier in their life and live longer with higher quality of life. Like, there's a lot of stuff that we know that just does not get implemented right now. And when I say what we know, I mean, at least to some extent, the scientific and medical community. And so there's a bit of a breakdown in communication. There's a lot of emphasis on focus on, we need to develop new cancer medications. We need to develop new drugs to treat whatever disease. about 50% of cancers actually are due to causes that are preventable by lifestyle modifications. So, right, these are cancers that are due to whether it's smoking or diet or exercise or various types of infections or sun exposure. About half of all cancers. In comparison, if you were to develop a drug that was just eliminated half of all cancers, that would be like an earth-shattering discovery. But we basically already have that discovery in the form of we know if you do these things, if you stop doing these things rather, you'll cut your risk of cancer in half. And so it's an interesting contrast where we're putting all this effort on like progress and development of new information. We need to put a lot more emphasis on implementing what we already know. And so part of the way to do that, and you can do this challenge, anyone who's watching, the only way you're going to get good at communicating information is to practice. You don't have to have a PhD, you don't have to be a doctor, you don't have to be in the healthcare field yet at all for you to do this, right? Because you can look up resources from the WHO or the CDC or research papers, and you can find something cool that people would benefit from learning. And so what my goal for you is just to start to practice. You'll be really bad at it first. I was really bad at first. I've since deleted many of my early videos. But that's the only way you're going to get better. A couple tips if you do want to try this, even with your own private social media network. Video format is more effective, particularly short video format. Bright lighting is important. You don't need a ring light. You can just face a window. Write down what you're going to say ahead of time. Your clickbait for the first few seconds is important. I wish it wasn't, but you kind of have to be a little bit extreme to get people to keep watching. Keep it short to like just a couple key points, which will lend itself to a shorter video duration, and then copy videos that are successful. You can do this with any number of topics. I'm going to skip over the influenza here. We're entering flu season, by the way. Important to get vaccinated this year. But some topics that you could do, there's a bunch. Why it's important to finish your full course of antibiotics so we don't get antibiotic resistance. Why heart health at middle age is one of the best predictors of cognitive health in older age because of the vascular blood supply to your brain, which keeps it healthy. How much your risk of heart disease goes down if you do some form of exercise for 15 minutes a day, right? Each of these videos, I guarantee you, people who you're connected with on Instagram or Facebook or TikTok or whatever platform it is, your friends and family could benefit from any of this information. And you would probably learn something by making one of these videos. So my challenge to you is just go ahead and do it. It's going to be bad, but the only way to get good is to test it. And so Voltaire has a quote here, or attributed to Voltaire. I'm not sure where it originally came from, but the best is the enemy of the good. If you hold out for, oh, I don't have a PhD in the subject, I don't have professional camera equipment, so I'm not going to make a video at all. That's not the right mindset for growth. You just basically need to try it, see what works, keep doing what works, stop doing what doesn't work. And I think you'll find it to be both rewarding and you could have a real impact on someone's life. So you should give it a shot. Okay, with that, we can go into any questions about anything, ranging from PhD, getting to applications, to jobs in science, to COVID questions, to COVID science and health media.
Okay. There's a question about from Ria Tabari. She was saying, asking, why is the mRNA mechanism of the vaccine so novel for the vaccines being developed? Is this not a common mechanism that's used?
That's a great question. And so the reason why mRNA is the first candidates to market here is it's the fastest to develop. So basically, as soon as we sequenced the first viral strains, coming out of China, you can just take that sequence for the spike protein, throw it into an mRNA vaccine, and you have vaccine material within a couple of weeks, basically. That's faster than producing mock non-infectious viruses. That's faster than producing proteins. And so the reason we don't have it traditionally for other diseases is we have well-established methods of protein-based and viral-based vaccines for other diseases, which work really well. And there hasn't been a sufficient impetus really to test out an mRNA vaccine to make the investment to see if it would work, basically until this time. I think it probably would have happened naturally anyway over the next couple of years. In fact, the companies are concurrently investigating mRNA vaccines for other things, including like cancer. So I think it was going to happen. It's just a relatively new format and only now has been worth the investment. Like its comparative advantage of speed is not a very big advantage for influenza because we already have a flu vaccine. But getting the first vaccine to market for COVID-19 is a huge advantage here. And so mRNA has an advantage in that field.
There's a question from Emma Bryant. How will the vaccine be dispersed as far as like the risk groups? Who will get access to it?
Yeah, I mean, I have a general answer. I actually don't know enough about the actual logistics of how it's going to work. The two we have so far, Pfizer's requires minus 70 to minus 80 degrees Celsius storage. I think that may change, but that's what it is for now. Moderna's can be stored at minus 20. And so Pfizer's will probably be in big, relatively urban centers who have those freezers. Moderna's may be more appropriate for rural. Now, in each of those cases, though, they're going to be rolling it out first to people who have some of the comorbidities that increase risk of severe disease with COVID-19, having obesity, being older, maybe having some respiratory conditions. There's a long list of comorbidities that increase your risk of severe COVID-19. Those people will get it first. And presumably also healthcare workers and maybe grocery store workers and people who have high rates of exposure to other people. When we were looking back at the diagram of how spread actually works, some of those people who spread it to 5 other people, maybe it's not because they're actually producing a lot of virus. Maybe it's because they work in the checkout lane at the grocery store and they just see hundreds of people per day or something. And so it would make sense to vaccinate those people first. But I don't have a very good idea of how this is going to happen. Like, there's not a database of people who have comorbidities. Like, I don't know whether it's going to be people's primary care physician who's recommending, like, yes, my patient has high risk for COVID-19, they should get a vaccine or something. But some people don't have primary care physicians, but definitely do have comorbidities. In fact, the people who don't have primary care physicians are probably the highest rates of having comorbidities, if I had to guess. And so I just don't have a good idea of how that's going to work. It'll probably be self-reported to some extent because I don't really know how it would work otherwise, but I would like to learn more about that.
Okay. With that being said, there was a lot of questions, but overall, I think it was a great productive call. We'll end the call right here, but I want to thank you for taking the time for speaking with me and all of us who came through. And a couple of announcements before we go. This call will be posted right after we get done. So if you join late or if you want to watch it again, this will be posted on the internet. And really quickly, I'm going to share my screen. You can follow us on Instagram at @officialclubmed. So if you like this call and this was your first time calling, we have calls every single week, and we've also had calls in the back, so you can always go watch those calls. We have calls ranging from DOs, MDs. This was our first PhD call, so that was amazing. But we'll continue to have a broad call so everybody finds something that they like. And secondly, we run this calls free of cost for anybody. With that being said, we still have costs that are associated with the program, such as emailing. If you want to sign up, if you go in the link of our bio, we have a weekly reminder set up where you will get an email on who the call is going to be, and you'll get that ahead of a couple of days so you know when the call is coming. You can go ahead and sign up for that under the link in the Instagram bio. If you would like to, if you can donate in any shape or form, any amount, we appreciate. Our Venmo and Cash Apps are up on the screen. And that's it. Thank you so much, guys. Thank you, Dr. Knock.
Okay, yeah, thanks for having me.